You are welcome, ladies and gentlemen. This is the Introduction to Cell Biology MIM Institute Series Lecture number 2. Today we are going through membranes. What are membranes? So I have with me the following subtopics to briefly discuss, and that includes general membrane function, membrane composition, phospholipid bilayer, membrane structure, post-translational modification, membrane fluidity, diffusion in membranes, movement of ions and molecules across membranes, transport across membranes, the Nernst equation. Remember, some of these subtopics will only be talked about in passing and they won't go into so much details. So here are some functions of the membrane, not all, but just some, ladies and gentlemen. Function number 1 is it enables ion and concentration gradients. Function number 2, it provides a diffusion barrier. Function number 3 is it separates one area of the cell from another. Function number 4, it concentrates membrane-associated molecules. So the following are the few compositions in the membrane, and they include cholesterol, a symmetric structure, negative phospholipids, PG, PS, PI, and neutral phospholipids, among which are PC, PE, and sphingolipids. Lipids include a variety of molecules such as triglycerides, phospholipids, and cholesterol. Lipids are biological molecules that are soluble in certain organic solvents. Whether or not something is a lipid is operationally defined by the solubility. The major type of lipid in the membrane is the phospholipid. They are phospholipids because they all contain a phosphate diester. Membranes separate one part of a cell from the other. Proteins, other molecules can be localized in the membrane, thus creating two-dimensional diffusion, which is more efficient than three-dimensional diffusion in a solution. It creates concentration gradients across the membrane by pumping specific molecules out of cell and by allowing specific molecules into the cell. So the commonest phospholipids are the neutral lipids, and these include phosphatidylglycerol, phosphatidylserine, phosphatidyl inositol, lecithin, phosphatidyl lethanolamine. The name of the phospholipid is dictated by the head group, and the tail are attached through the phosphate diester. The sphingomyelin. Under this we have glycolipids, cholesterol. Glycolipids are derived from sphingosine but have a sugar unit such as glucose or galactose attached instead of the colon unit. The sugars point out from the cell surface and are involved in cell-to-cell recognition. Cholesterol is an essential component of mammalian membranes. It is obtained from the diet or can be synthesized from acetyl-CoA. The phospholipid bilayer. This consists of two layers, tails inside, heads outside. The hydrophobic part is 30 angstrom thick. Phospholipids are detergents. They are hydrophobic part. The fatty acetyl and a hydrophilic part, which is the head. So the phospholipids associate with each other through hydrophobic interaction, forming two layers or leaflets of phospholipid. This buries the hydrophobic fatty acid tails and exposes the polar part or the head to water because of the thick layer of hydrocarbon. Any molecule that may try to penetrate the bilayer must pass through this hydrophobic region. For polar molecules and ions, this is very difficult because they must lose the strong interactions they make with the water in order to pass through the layer. The membrane structure. Membranes are symmetric. Membrane spanning segments and lipid modification, fatty acid acylation and prenylation anchor proteins in a fluid bilayer, single fluid mosaic model. The membrane is asymmetric because the inner and outer leaflets can have a different lipid composition and contain different proteins. Proteins can be associated with either side of the membrane, or they can pass through the membrane using membrane spanning segments. The functional part of the protein can be on the cytosolic side, the external side, or even in the membrane itself. A common structure for spanning a membrane is an alpha helix. But there are examples of sheets spanning a membrane. It takes about 20 amino acid residues arranged in a helix to span to a 30 angstrom hydrophobic interior of the bilayer. In the later lectures, when we discuss the proteins, amino acids, we shall see just how exactly this applies, and we should be able to see the relationship between proteins and phospholipid bilayer, just how they help in signal transduction, conduction of molecules, in molecular recognition of cell to cell. Peripheral membrane proteins can be removed from cells by means of salt solutions or low pH solutions because these are able to disrupt ionic interaction. Integral membrane proteins cannot be removed by means of detergents. To distinguish between integral and peripheral membrane proteins is simply based on mode of operation rather than the structure. So you shall have more of such distinctions under biochemistry series of lectures. But under cell biology, we don't go into all those details in this under biochemistry where we do such works very well. So here is how it works, the distinction based on operation rather than structure. All proteins that pass through the membrane one or more times will be integral membrane proteins, but not all integral membrane proteins will pass through the membrane. It depends on whether or not the protein can be removed by salt or low pH washes. Peripheral membrane proteins associate with the membrane, or more usually with integral membrane proteins. Post-translational modification can affect membrane association by prenylation or fatty acylation. Glycoproteins and glycolipids on the exterior surface of the membrane have carbohydrates attached. Some proteins can be post-translationally modified by the addition of prenyl groups. Prenyl groups are long-chain unsaturated hydrocarbons that are intermediate in isoprenoid synthesis. Glycoproteins and glycolipids have complex sugar residues attached. Since they are attached in the ER and Golgi compartments, the sugar coating will point outward from the cell, will be on the outside surface of the membrane. Membrane proteins as well as phospholipids and glycolipids are embedded in the lipid bilayer and move around the plane of bilayer very rapidly. Membrane fluidity. Increasing fluidity makes electron diffusion faster. Fluidity increases with increased temperature, increased content of short-chain fatty acids, and increased content of cis fatty acids. Cholesterol increases the fluidity of membranes that are not very fluid, but decreases the fluidity of membranes that are already fluid. The membrane is a dynamic assembly and things are diffusing rapidly in the plane of the bilayer. The middle layer of the bilayer has been linked to olive oil. As with oil, cooling of the lipid bilayer will cause the hydrocarbons to become more ordered, structured. The side chains pack closer to each other and the fluid of the membrane is lower. Things that disrupt the ability of the side chain to pack in the irregular fashion make the membrane more fluid. These include high temperature, lipids with shorter chains, and lipids with cis double bonds. The shorter lipids and cis double bonds cause occurrence of holes, that is, packing defects. Cholesterol has a funny effect on the membrane fluidity because of its shape. Cholesterol prevents long-chain fatty acids from packing close to each other. However, cholesterol is just the right size to pack into the defects caused by cis fatty acids. In a membrane, like most mammalian membranes that contain significant fatty acids, adding cholesterol will cause the membrane fluidity to decrease. Diffusion in membranes. Lateral diffusion is in the plane of the membrane, and transverse or flip-flop diffusion is perpendicular to the membrane, that is, through the membrane. Lateral diffusion is 2 dimensions, is fast, and transverse diffusion is slow or non-existent except for gases such as carbon dioxide, ammonia, and hydrophobic uncharged small molecules such as cholesterol. To diffuse rapidly in the plane of the membrane, lateral diffusion, that is, a molecule must simply move around in the lipid environment, that is, including the polar head groups. This being the same in the same environment, it needs not to change how it interacts with other phospholipids or with the water. Lateral diffusion can be slowed or even prevented by interactions between membrane proteins and cellular cytoskeleton. This specially restricts a plasma membrane protein to be localized in an environment. Desolvation must take place, or there must be a loss of interactions with water by ions and proteins. Hence, this way they'll change sides or traverse the membrane, or they'll go through the membrane, thus going through that hydrophobic region of the phospholipid bilayer. Now, this one you must know that it's extremely difficult. Hence, ions and proteins do not move through the membranes by themselves. Small molecules such as carbon dioxide, ammonia, but not the ammonium ion, and water can diffuse through membranes. However, most other small molecules pass through the lipid bilayer very slowly, if at all. Thus, there must be mechanisms by which to do this. Now, let us look at movement of ions and molecules across membranes. This requires the participation of protein transporter molecules move spontaneously toward lower concentration, that is chemical gradient, and opposite charge, that is electrical gradient. Moving in the opposite direction requires the input of energy. There are two factors that influence the movement of ions and molecules through a membrane. These are concentration gradient across the membrane, also called chemical potential, and the electrical potential of the membrane. Concentration gradient, also known as chemical potential, exists if the concentration of a given molecule or ion is different on the two sides of the membrane. Normally, cells maintain a slight excess of negative ions inside the cell. This costs energy, but this uneven distribution of charge across the membrane results in a membrane potential or electrical potential. The membrane potential is negative, indicating that the inside of a cell is negatively charged as opposed to the outside, which is positive. It has a normal value of negative 0.06 volts or negative 60 millivolts. As long as the membrane potential is maintained, it will affect how ions move. Moving an ion toward the opposite charge will be easier than moving the ion toward the same charge. The membrane potential and the concentration gradient can reinforce each other, or they can be in opposition to each other. The total force tending to move a molecule or ion through a membrane is called electrochemical potential. When the concentration gradient and the electrical potential work to oppose each other, the stronger effect wins. If someone forces you to get quantitative, this may be physiologist rather than a biochemist. Let's go to the next section, that is transport across membranes. Facilitated diffusion, that is via a channel. Molecule moves down its electrochemical gradient. Active transport by means of a pump. Molecule moves up its electrochemical gradient, requires energy input. Pumps use energy, that is usually ATP hydrolysis. Sodium ion is high on the outside compared to the proportion of potassium ion, which is high on the inside. Because membranes are impermeable to most molecules, there must be a transporter or a protein in the membrane to help molecules or ions move through it. These are also called channels because they behave like selective holes in the membrane. The transporters are selective because they usually allow only one type of ion to pass through the membrane. For example, the calcium channel that releases calcium from the endoplasmic reticulum releases only calcium but not other divalent cations. What distinguishes a channel from a pump is that the movement of ions through a channel does not require energy input. The channels can also be gated. This means that something causes the channel to open. For example, a ligand-gated channel opens when a specific ligand binds to a receptor in the membrane. Vogt-gated channels open and close in response to changes in the membrane potential. Pumps move ions and molecules up their electrochemical gradient. Pumps require energy, usually in the form of ATP hydrolysis. Sodium potassium ATPase is an example of a pump. Cells maintain a higher concentration of potassium inside the cell than they do outside the cell. Sodium is maintained lower inside, high outside. Sodium potassium ATPase pumps 3 sodium ions from inside the cell to the outside. This is the unfavorable direction because the sodium ion moves from low concentration to the higher one and against the membrane potential. At the same time, pumps 2 potassium ions from outside the cell to inside against the concentration gradient, but with the electrical gradient. Both ion movements are unfavorable to the transport process, requires energy. The pumps work by changing their structure and binding characteristics during the cycle of ATP binding, hydrolysis, and release of ADP and a phosphate molecule. Exactly when and what triggers these changes will vary from pump to pump, but the essential feature is that there must be a cycle that moves the binding site from one side of the membrane to the other, while it also changes the affinity for the ligand. When it faces outside, the sodium binding site must have a low affinity for sodium. This allows sodium to be released where the concentration is high. The potassium binding site will have a high affinity for potassium when it faces outside. This allows potassium to be taken up outside where concentration is low. During the movement to the other side of the membrane, this doesn't happen the same time for the potassium and sodium sites. The affinity for substrate changes. When it faces inside, the sodium binding site has a high affinity so that it can take up sodium at the low concentration inside. The potassium site that faces inside has a low affinity so that it can release potassium at the high concentration inside the cell. Ion gradients can also be used to transport other molecules. During digestion, glucose is concentrated at the intestinal epithelium, moves from low concentration inside to a high concentration inside. The Nernst equation will not be discussed, and as we said, some portions of this lecture will only be looked at in passing. Here ends the lecture. Remember to subscribe.